Fruits and vegetables are produced during peak seasons, but due to lack of preservation and storage facilities, the market becomes overstocked during such periods and get rotten before reaching the final consumer. Many processing techniques can be employed to preserve fruits and vegetables. Drying and dehydration is one of the most important operations that are widely practiced because of the considerable saving in packaging, storage, and reduced shipping weight, etc. Osmosis is a physical phenomenon that has been extensively studied in various disciplines of science and engineering. Osmotic dehydration has diverse applications in fruits and vegetables. Thus, it can be used to decrease the post harvest losses. Being a simple process, it facilitates processing of fruits and vegetables with retention of initial fruit characteristics such as color, aroma, texture, and nutritional composition. Since no preservative was used, it does not adversely affect the human body. This process could be used on small scale for development of self-entrepreneur and home scale industries. So let's talk about the theory behind the osmotic dehydration in food processing. Hello everyone, welcome to our YouTube channel. If you are new to here and want to learn about the food processing technology, please hit the subscribe followed by the bell. Today we are going to talk about osmotic dehydration and its applications. Osmotic dehydration is a phenomenon of removal of water from lower concentration of solute to higher concentration through a semi-permeable membrane results in the equilibrium condition in both sides of membrane. Osmotic dehydration, also called the dewatering impregnation soaking process, is a water removal process that involves the soaking of fruits and vegetables in a hypertonic salt or sugar or in a combined solution to reduce the water content while increasing the soluble solid content. This is a multi-component diffusion process. In this process, water flows from fruits or vegetables to the solution, and along with the water, some components of fruits and vegetables such as minerals, vitamins, fruit acids also move towards the solution. The sugar and salt migrate towards the fruits and vegetables. The process of complete osmotic dehydration is shown by the diagram. When the raw material is placed in a concentrated solution of soluble solids with higher osmotic pressure and lower water activity, three types of counter mass transfer phenomenon are happen in osmotic dehydration process. First one is water diffuses out of the product to the solution at a faster rate initially and slowly afterwards. Second one is the solute transfer from the solution to the product. It makes thus possible to introduce the desired amount of an active principle, a perspective agent, any solute or nutritional interest, or a sensory quality improvement of the product. And the third one is leaching out of the own solutes, which is quantitatively negligible when compared with the first two types of transfer, but essential with regard to the composition of final product. The two main solutes used in osmotic dehydration are sugar and brine. Osmotic dehydration found wide application in the preservation of food materials since it lowers the water activity of fruits and vegetables. Osmotic dehydration is preferred over other methods due to their color, aroma, nutritional constituents, and flavor compounds retention value. Osmotic dehydration as a mass transfer process is affected by different factors. They can be divided under three categories. They are pre-treatment factors, product-related factors, osmotic solution-related factors. Pre-treatment conditions before osmotic dehydration process affect the product inherent integrity, which has an effect on mass transfer process. The cell membrane permeability strongly affects the dehydration rate. Good membrane permeability ultimately will lead to more rapid osmotic dehydration. Unfortunately, the rate of mass transfer during osmotic dehydration of plant foods is generally slow process. The cellular membrane exerts high resistance to transfer and slow down the osmotic dehydration rate. Thus, there has always been a need to develop supplementary techniques to enhance the mass transfer without adversely affecting the quality. Therefore, the partial damage of cell membranes using different pre-treatment methods can be advantageous for acceleration of mass transfer process. These techniques include blanching, peeling, coating, freezing and thawing, high hydrostatic pressure processing, pulse electric fields, ultrasound treatment, vacuum impregnation, and ohmic heating. The shape of the solid material is an important factor because the moisture loss and solids gain decrease with an increase in sample size. If the solid is bigger in size, it will dehydrate more slowly because the length of the diffusion path is greater and less surface area is available for solid exchange. Hence, the solid will enter the sample at a slower rate. During osmotic treatment, the semi-permeable cell membrane can change from partial to total permeability, leading to significant changes in tissue architecture. The loss of water during osmotic treatment resulted in cell shrinkage, leading to cell damage due to loss of contact between cell membrane and cell wall. Particularly, porosity of the raw material has a significant effect on shrinkage phenomena and mass transfer rates as well as rehydration ratio. Also, tissue structure plays a very important role not only in terms of the total porosity but also the size and shape distribution of pores and their interaction with external liquid. The selection of the solute must consider the following main factors such as temperature of osmotic solution, concentration of osmotic agent, type of osmotic agent, agitation process during osmotic process, osmotic solution, and food mass ratio. Fruit sample has a porous structure so that high temperature would also release the trapped air from the tissue resulting in more effective removal of water by osmotic pressure. The solute choice and concentration depends on several factors, namely the effect of organoleptic properties, solid solubility, cell membrane permeability, its stabilizing effect and cost. The two most common solute types used for osmotic treatments are sugar and salt. Researchers have revealed that mixed Sucrose salt solution gave greater decrease in product water activity than pure sucrose solutions, although the water transport rates were similar. The pH of the solution can also affect the osmotic process. Acidification increases the rate of water removal by changes in tissue properties and consequential changes in the texture of fruits and vegetables. There are advantages of osmotic dehydration. Fruit dehydrated by osmosis is not subjected to high temperature or an extended time. Therefore, heat damage to color and flavor are minimized. Use of sugar as the osmotic solution prevents much of the loss of fresh fruit flavor, which is commonly found with conventional air drying or vacuum drying methods. Also, high concentration of sugar surrounding the fruit pieces prevents discoloration of fruits by enzymatic oxidation. Osmotic dehydration alone cannot provide longer shelf life and stability to the final product. Combination drying is the best way to reduce the energy consumption and increase the throughput and improve the quality. Hence, osmotic dehydration processes are being used in combination with convective, freeze, microwave, vacuum or infrared drying steps. Here you can see a process line that osmotic dehydration process is practiced. So, these are the basic things that you should know about the osmotic dehydration. You can search for the details through research papers and books. If you found this video helpful, please give it thumbs up and subscribe for videos like this. Hope to catch you in next time and thanks for watching.